Hello everyone, myself Dr. Satish Maurya. Welcome back to the next module, that is Transition to Flowering Part 2. Besides day length changes, leaf senses temperature fluctuation and regulate flowering in response to such fluctuations. Then there is an autonomous pathway of flowering which brings about flowering in a photoperiod or low temperature independent manner. The plant hormone that is known as gibberellic acid or GA in short also induces flowering through interactions with the photoperiod signaling pathway. Finally, there is an age dependent pathway which enables the plant to respond to flowering cues only when the plant reaches the adult vegetative phase. Let us see how these plant pathways regulate flowering time. Before moving to the actual part, the whole module is divided into three major parts. So the first part is, or the first objective of this module is to see how temperature fluctuations affect the induction of flowering. The second one is to understand how the autonomous and GA dependent pathways regulate flowering time. And coming to the third objective of this module, which is nothing but the age of plant, how the age of plant is important to entering or for entering the flowering phases. Coming to the first objective, which is nothing but vernalization pathway. So first question is, what is vernalization? Vernalization, or in many plants, requires an extended cold period before they flower in long days when the temperature is warmer. This pre-flowering cold exposure is called vernalization, in which not only the cold temperature but also the duration of the cold exposure is monitored by plants. Vernalization was first observed in temperate cereals such as wheat. In these annual crops, vernalization is required to promote the transition to flowering, and without vernalization, the crops show only vegetative growth. Winter annual ecotypes of Arabidopsis also require vernalization to promote flowering. In Arabidopsis, a Matchbox transcription factor, flowering locus C, that is FLC, is a potent flowering repressor of flowering. It is active during the vegetative stage and represses the flowering inducers such as FT, which is nothing but flowering locus T, FD, which is nothing but flowering locus D, and SOC1, which is nothing but suppressor of overexpression of constans 1 in the inflorescence meristem. Another gene known as Frigida, which is in short known as FRI, encodes a plant-specific scaffold protein that functions as an assembly platform and forms a large protein complex with its associated partners and active domain or active chromatin modifier, so which is highlighted here. The FRI complex brings about transcriptional activation of FLC. Exposure to prolonged cold conditions leads to sequestration of FRI from the FLC promoter. This prevents the FRI induced activation of FLC. The expression of FRI during embryogenesis restores FLC activity in the vegetative stage. FLC silencing also involves a long non-coding antisense transcription of FLC gene called cool air, which is also a part of the cold sensing mechanism. Induction of these antisense transcriptions on cold exposure leads to a transient reduction in sense transcription of FLC. Further, prolonged cold exposure results in FLC silencing by repressive chromatin modifiers, leading to a repressed chromatin state at FLC locus. Stable repression and histone modification of FLC is carried out by several genes such as vernalization, which is nothing but VRN1 and VRN2. and vernalization insensitive or VIN3. VRN1 encodes a scaffold protein and VIN3 encodes a chromatin remodeler protein called plant homeodomain finger, that is PHD protein. Win3 expression increases after transfer to cold and then decreases rapidly after transfer to warm conditions. Win3 heterodimerizes with a related PHD protein called VRN5 and brings about the formation of PHD PRC2 complex. The core component of PRC2 complex includes several H3K27 methyl transferases and core proteins, one of them being the zinc finger protein vernalization 2, that is VRN2. The PHD PRC2 complex progressively silences the whole FLC locus through H3LYS, that is lysine 27 trimethylation, and the number of FLC silenced cells increases with longer period of cold. Hence, the extent of FLC silencing during the cold exposure serves as a quantitative measure of the duration of the vernalization period. After return to ambient temperature, cold-induced chromatin-based FLC silencing is maintained, and this epigenetic memory of winter cold enables floral induction during long days and ambient temperature in spring. During reproduction, the repressed chromatin state at FLC is transmitted to the zygote and early embryos. This is reset but is reset during subsequent embryo development, necessitating the winter exposure in the each generation in order to flower. Further, in contrast to vernalization mechanisms, smaller changes in ambient temperature also regulate the timing of floral transition. The ambient temperature pathway mainly acts at moderate temperatures between 16 degrees Celsius to 27 degrees Celsius to regulate the flowering. In Arabidopsis, shows late flowering when grown under short days and lower ambient temperature, and early flowering when grown at higher ambient temperature. Hence, late flowering in short days can be overcome by growth at higher temperatures. This difference is due to direct thermoregulation of FT gene expression in leaves, while the major pathway known to upregulate FT in the photoperiod pathway, this pathway does not play a role in the thermal acceleration of flowering. Ambient temperature signaling is mediated by matchbox TF short vegetative phase, that is SVP. Another matchbox TF flowering locus M, that is FLM, forms a heterodimeric complex with SVP. FLM shows sequence similarity to FLC but does not contribute to the vernalization requirement. The FLM locus generates two major splice variants, that is FLM beta and FLM delta, which exert opposite effect on flowering. SVP shows preferentially binding to FLM beta at low ambient temperature. This brings about nuclear translocation of SVP, where it represses the FT expression and delays the flowering. The nuclear localization of SVP decreases as temperature increases. SVP preferentially interacts with FLM delta at high ambient temperature and causes rapid degradation of SVP via ubiquitination, hence removing its repression of FT. SVP mediated control of FT gene expression represents a mechanism used by the plant to adjust the timing of flowering under fluctuating temperature conditions. The phytochrome B, that is PHYB photoreceptor, has been shown to function as a temperature sensor in addition to its role as a red light sensor. It participates in temperature perception through its high ambient temperature dependent reversion from the active PFR to the inactive PR state of light. The PFR form of 5B represses the TF phytochrome interacting factor 4, that is PIF4, through the photoperiodic promotion pathway under long days. However, 5B reversion at high ambient temperature releases its repression on PIF4 and brings about PIF4 mediated induction of FT and the thermal acceleration of flowering under short days. Besides 5B mediated de-repression of PIF4, other thermosensory pathways also play a role in PIF4 mediated FT expression. Exposure to high ambient temperatures leads to the eviction of the histone variant, that is H2AZ, present in nucleosomes, which impedes access to the transcriptional machinery due to the light chromatin conformation. Removal of H2AZ from temperature sensitive promoters of the flowering gene FT makes it more accessible to specific transcription factors. This enables PIF4 to bind to the FT promoter at high ambient temperature to accelerate flowering. Another pathway that regulates PIF4 expression at high ambient temperature is mediated by early flowering, that is ELF3, which is a component of the evening complex or EC of the circadian clock. At low ambient temperature, ELF3 as a part of EC represses the expression of PIF4. At higher ambient temperature, ELF3 forms nuclear condensates, due to which EC inhibition of PIF4 is relieved and PIF4 transcription brings about PIF4 mediated FT expression. Hence, PIF4 mediated FT expression enables the plant to regulate flowering at high ambient temperature during short days. A third pathway, the autonomous pathway, promotes flowering in a photoperiod or low temperature independent manner. This pathway was identified by analyzing mutants that are still responsive to photoperiodic cues and to vernalization, but yet show late flowering. The autonomous pathway leads to FLC repression and the promotion of flowering. The gene associated with this pathway are either chromatin associated protein or RNA binding repressor factor. For example, flowering locus T, FLD, is involved in histone modification, while flowering locus CA, that is FCA, flowering locus PA, that is FPA, and flowering locus Y, that is FY, contain putative RNA binding domains and play a role in RNA processing. FCA mediated repression of FLC is the best understood autonomous pathway. FCA is a nuclear RNA binding protein that interacts with FPA and FY. FLD encodes a histone dimethylase that removes H3K4ME2, that is active chromatin, and increases the H3K27ME3, that is inactive chromatin, at FLC chromatin. FCA, FPA, and FY activities also promotes proximal splicing and poly-A of cool air and antisense transcript of FLC gene. which is important for FLC repression. FCA and PFA mediated co-transcriptional RNA processing events seems to be involved in regulating the chromatin modifying activity of FLD. Plant shows morphological and physiological changes during transition from juvenile to adult phase and from the adult to flowering phase. During the juvenile vegetative phase, Arabidopsis is not responsive to the flowering cues, and it is only when it reaches the adult vegetative phase the flowering is induced. Age-dependent flowering initiation occurs through FT expression, which is regulated by microRNAs, that is miRNAs, that is miRNA 156 and miRNA or microRNA 172. These miRNAs are known to cleave the transcripts of squamosal promoter binding proteins like SPL genes, which codes for plant specific TFs that regulates many aspects of development including flowering time, and miRNA 172 and 156 are in turn positively regulated by the transcription factors they target, indicating a negative feedback loop between the miRNAs and their targets. High level of microRNA 156 levels are seen in young plants, which brings about the cleavage of SPL transcripts and renders the plant incapable of responding to flowering cues. MicroRNA 156 levels decreases with age, most likely due to sugar accumulation arising from photosynthetic activity, and this allows the SPL3, 4 and 5 to accumulate, which binds to the FT promoter and activates its transcription to promote the flowering. MicroRNA 156 also targets the SPL9 and its homologous SPL10, which are required for the miRNA, that is microRNA 172 transcription. Hence, cleavage of SPL9 by microRNA 156 leads to the inhibition of microRNA 172 expression during the juvenile vegetative phase. MicroRNA 172 expression increases with age, and microRNA 172 targets includes the TF called target of early activation tag, that is TOE, which belong to the AP2 family of transcription factor. MicroRNA 172 forms an RNA duplex with a sequence near the 3' end of the coding region of the targets. The removal of these repressors promotes the expression of FTs in the adult phase. The gibberellin, that is GA pathway, plays an important role in the regulation of flowering under both short day and long day conditions. Expression of a key GA biosynthesis enzyme, that is G820 oxidase, increases when plants are transferred from short day to long day, and this leads to higher GA level and early flowering under long days. The SVP protein, which is an inhibitor of flowering induction, is also seen to inhibit G20 oxidase, thus bringing about lower GA levels. Besides GA biosynthesis, regulation of flowering time is also mediated by GA signaling. DELA proteins are negative. So DELA proteins are negative regulators in GA signaling as well as flowering time. Since DELA proteins possess no DNA binding domain, they modulate gene expression by interacting with other proteins. DELA have been shown to bind to CO protein, leading to an inhibition of CO activity. GA synthesis leads to proteasomal degradation of DELA proteins, which brings about an increase in CO protein accumulation under long days and induction of flowering. Under short days, DELA proteins interact with FLC to enhance the protein stability and promote the repressive transcriptional activities. The repressive complexes blocks the expression of FT until increased GA signal triggers the degradation of DELA. Similarly, under fluctuating ambient temperatures, DELA proteins interact with the FT activator PIF4 and prevent PIF4 from binding to the FT promoter. The GA-induced DELA degradation then allows PIF4 FT interactions to promote flowering at high ambient temperature. The squamosa promoter like SPL genes are also inhibited by DELA expression and prevents the transition to flowering in the age-dependent flowering induction pathway. Hence, the GA signaling pathway interacts with the other flowering time pathways in regulating flowering induction. Hence, besides the photoperiodic pathway which has been dealt with within the previous module, there are several other pathways that are involved in the induction of flowering, such that it occurs when environmental conditions are favorable. Thermal regulation of flowering occurs in response to exposure of plant to extreme cold temperature for a long period, which is also known as a vernalization, or in response to changes within the ambient temperature range for short durations. Besides these pathways, flowering can be induced in the absence of environmental cues through an autonomous pathway, or the plant hormone GA may also trigger flowering. Finally, age of the plant is important in regulating the transition to flowering, and this is mediated through an age-specific expression of microRNAs and their targets. A plant will experience inhibitory and promotive signals from each of these pathways, which ultimately regulates when the plant flowers. The time of flowering affects the reproductive success of a plant through optimizing the time for pollination or seed dispersal. In outcrossing species, this is of particular importance, as flowering time needs to be coordinated with other plants of the same species in their vicinity, and for insect pollinated plants with the activity of their pollinators. The flowering time pathways converge to regulate the expression of FT and SOC1, which are integrators of the various flowering signals and lead to the formation of floral meristem. These will be dealt within the next coming modules. Thank you.